My name is Xingzhen Yang. I'll introduce the paper submitted to iMeta. The title is HLF and PPARa axis regulates metabolic-associated fatty liver disease through extracellular vesicles derived from the intestinal microbiota. Metabolic-associated fatty liver disease, MAFLD, is the most prevalent chronic liver condition globally, affecting approximately 25% of the general population and over 50% of individuals with metabolic dysfunction. However, its pathogenesis remains incompletely understood. The multiple-hit hypothesis is currently the most widely accepted mechanism underlying its development. Extensive research has confirmed the significant role of intestinal lipid absorption in MAFLD. In recent years, extracellular vesicles, EVs, as crucial mediators of intercellular communication, have become a focal point of research. Recent studies have demonstrated that orally administered EVs can protect the liver from damage and improve metabolic conditions. These findings offer promising prospects for the prevention and treatment of metabolic disorders based on gut microbiota-derived EVs, providing theoretical support for investigating the regulatory role of the gut in MAFLD. The highlights of this study are as follows. First, intestine-specific deficiency of hepatic leukemia factor, HLF, ameliorates MAFLD. Second, HLF regulates lipid metabolism through peroxisome, proliferator-activated receptor alpha, PPARa. Third, the HLF and PPARa axis modulates the gut microbiota, influencing the composition of microbiota-derived extracellular vesicles, FEVs. Fourth, the conjugated bile acid taurochenodeoxycholic acid, TCDCA, is a key regulator in FEVS-mediated improvement of MAFLD. Initially, single-cell RNA sequencing and bulk RNA sequencing of intestinal tissues from healthy and spontaneously fatty liver cynomolgus monkeys were conducted to identify the gene HLF, which influences intestinal lipid absorption. Subsequently, intestine-specific HLF-knockout heterozygous and homozygous mouse models were established, demonstrating that intestinal HLF can regulate hepatic lipid metabolism. Downstream target genes of HLF were screened through intestinal transcriptome sequencing. And a mouse model using the PPARa inhibitor, GW6471, confirmed that inhibiting intestinal PPARa ameliorates MAFLD. Given that EVs are essential mediators of intercellular communication, we isolated gut microbiota-derived EVs and validated at both cellular and organismal levels that these EVs alleviate MAFLD by inhibiting ferroptosis. Furthermore, lipidomics and functional experiments revealed that the HLF and PPARa axis regulates gut microbiota levels related to bile acid metabolism, identifying TCDCA as the key molecule through which FEVs exert their lipid-lowering effects. This study underscores the importance of intestinal HLF and PPARa in lipid metabolism and MAFLD pathogenesis, highlighting the potential of gut microbiota-derived EVs and bile acid metabolism as therapeutic targets. The identification of TCDCA as a critical regulator offers new insights into the mechanisms underlying MAFLD and paves the way for innovative treatment strategies.